Hi, in this short video I will guide you how to make a simple visualization in Pymol and how to use it for molecule modeling applications and how to make different kinds of visualizations, graphical representations and so on. So when you open the Pymol, first of all you can change the color of the screen. So go to Display, Background, White. So as a standard, usually we make all the pictures in front of white background. Okay, so let's use the adenosine A2A receptor of human origin as a test molecule. So go to Fetch and enter the code from PDB. Okay, here we have the molecule. As you can see, it's a GPCR, typical G protein coupled receptor. So in Pymol, we use left mouse button for rotation, right mouse button for scaling, and middle mouse button for moving. There is another function of the wheel. If you scroll it back and forth, you will see that the molecule is sliced in parts. So usually it's used for different visualizations. For example, in basic mode, you see that ligand is lying inside the binding pocket, but to make better visualization, you can slice the view and cut out the front part. For this, use the scroll, and now you can open the way toward the ligand. Okay, so here we have the main part. All of the molecules that are loaded are indicated here, so you can click here on the name of the molecule and it will be hidden or shown. Here we have 5 buttons which control 5 different menus. The first A is action. Here we have different kinds of actions. For example, we can zoom our molecule, orient it, apply different presets, find polar interactions inside the molecule, align it with other molecule, and many other applications. Then we have also S menu from the word show. It's responsible for different representations. For example, we can visualize surface here, but to hide it we can use the third menu H, which means hide. The fourth menu L means label, and it controls different tools for labeling. And the last one, C from color, controls the coloring scheme. Okay, now let's visualize only the binding pocket and remove everything that is unnecessary. For this, go to Display, Sequence, have the whole sequence of our protein. So the color of letters represent the color of your molecule. Ligand here is called ZMA. So you can press on it and now you can manipulate the selected ligand with this menu seller. So each time when you select something, it will appear in the vertical menu here as the seller. So let's isolate it, extract object, and now we can always address directly the ligand by pressing this object. And as you can see, now if you press on it, the ligand appears and disappears. For convenience, let's rename it. Rename object and put as ligand ZMA. Let's change the color of ligand. Let it be magenta. And as you remember, it is widely accepted that the standard coloring scheme for elements should be used in molecular modeling applications. Okay, so now we have our ligand visualized in magenta color. To show only the residues around the ligand, we can go to Selection still, or you can press with left mouse button on the ligand and then go to selection, select, modify and expand by 5 angstroms residues. So it will select all the residues within 5 angstroms from ligand. Then make show sticks, then go back to selection, modify, invert within any. So now we inverted the selection, and everything that is far from the ligand than 5 angstroms is selected. So now click on hide everything, and now you have only ligand and its nearest environment. So this is one kind of visualization. And now we will apply one of the pre-generated presets to show the surface and shape of the binding pocket. For this, select again the ligand, expand the selection, and now when we selected the residues near the ligand and the ligand itself, we can go to selection, preset, ligand sites and transparent surface. As you can see, now we have the shape of the binding pocket, transparent, the solvent, the H bonding network, and the nearest residues. You can also modify the transparency. You can go to selection, preset, ligand sites, solid surface. This time we have a solid surface. You can visualize again the sticks. And now we will compare our target protein with the other protein of the same family, and actually the same protein, again adenosine A2A receptor from human, but bound to other ligand, to caffeine. For this, go to command line and fetch 3-RFM PDB. So as you see, it's again GPCR protein. We will first superimpose them by using command super. So the syntax is as following: super space, then indicate the second molecule that we are going to superimpose over the reference. And now we can center everything. Now let's hide the cartoon. So now we see that caffeine, indicated in blue, is superimposed over our initial ligand. And as you see, the position of these purine rings are similar. But let's also visualize the residues that surround the caffeine. For this, click on caffeine, go to selection, modify, expand by 5 angstroms residues, and then go back to modify, restrict to object. So only residues from the protein that is complex with caffeine will be selected. And now show sticks. They will be indicated in blue. So as you see, position of the residues is quite similar to the position of the same residues in initial protein, but with some minor differences. Okay. So if you like the view, you can execute command array. This command renders your view in a high quality picture, and then you can save it. Go to File, Save, Export Image as PNG, and here capture current display and click Save PNG Image. So you can use this picture directly in your manuscripts, in your posters, presentations, and so on. And I have to say that Pymol for many years is widely recognized as the golden standard software for molecular modeling visualizations. So I strongly recommend you to spend some time and master it. Thank you for your attention.